was a family in New Jersey, but originated from a town in Campania called Contursi. So finally, after many years of following the family, the pedigree, and trying to identify what was the genetic alteration that underlied the development of Parkinson's disease or Parkinsonism in this family, Polymeropoulos and colleagues, they identify this mutation. This, I'm showing here the sequence of 140 amino acid that is the sequence of the alpha-synuclein protein. And and Polymeropoulos and colleagues, they identify a single point mutation in position 53 with the substitution of alanine in position 53. So this paper came out, and we were all very surprised for many reasons. And perhaps the most important, the main reason was the fact that they talked about this protein and this genetic alteration, this protein called alpha-synuclein. And at the time, nobody had ever heard about alpha-synuclein in general, and in particular in Parkinson's disease field. So we were all completely surprised. And in fact, initially, there was just a bit of skepticism when people saw the paper and said, well, is this real? Is it true? Is it possible? But some an important confirmation that this mutation and the link between this mutation and Parkinsonism came only, I think, 1 or 2 years later, when a second mutation was identified in position 30 of the same amino acid sequence of alpha-synuclein by Kruger and colleagues. And at that point, we basically all started thinking, well, it seems to be pretty clear that mutations in this protein can cause a Parkinsonian syndrome. And since then, there have been other mutations that have been identified and linked to a Parkinsonian syndrome. And I'm listing this mutation in the bottom of this slide. But we also, again, in the late 90s, when the paper of Polymeropoulos came out, and then after that the paper of Kruger came out, we said, well, this is interesting, but could this be just a very rare association between alpha-synuclein and very rare genetic cases of Parkinsonism? And the answer to this question and to this concern came immediately after. And I think this is a classic example where, to go forward, we might need to go backward. You remember that yesterday we briefly mentioned and discussed the fact that in the 1912, Frederick Lewy described this intraneuronal inclusion called Lewy bodies and Lewy neurites. And I also mentioned yesterday that these inclusions are pathognomonic of Parkinson's disease. In other words, they are present in all patients with typical Parkinson's disease. So whenever you do a pathological analysis, a neuropathological analysis of the brain of patients with Parkinson's disease, you do find Lewy bodies. And in fact, a pathologist, a neuropathologist, will not make a diagnosis of Parkinson's disease unless he or she sees Lewy bodies and Lewy neurites in the brain of these patients. So now moving forward, right after the discovery of alpha-synuclein, it turns out that people developed antibodies against alpha-synuclein, and they stained the brain of patients with Parkinson's disease with these antibodies against alpha-synuclein. And in particular, Maria Grazia Spillantini in Cambridge was the first to develop antibodies and to use these antibodies in brain. And you see, 1 year after the paper of Polymeropoulos in 1998, she published this seminal paper in which she basically described and identified alpha-synuclein as a major component of Lewy bodies. Now, of course, why was this important? Not only this was important from the neuropathological standpoint, because we finally allowed us to characterize these inclusions much better, but it was an enormous step forward, because as I mentioned before, we were thinking at the time that maybe alpha-synuclein was only involved in rare familiar cases of Parkinsonism with rare mutations that I've described before. Now, the fact that Maria Grazia, and then of course many other people, neuropathologists, could see alpha-synuclein positive Lewy bodies in all patients with Parkinson's disease, it of course indicated very clearly that alpha-synuclein is implicated in the pathogenetic process that underlie the development of Parkinson's disease in all patients with Parkinson's disease. So it's not a rare mutation and a rare problem in specific families with this mutation. It's actually alpha-synuclein is a critical component of the development of the pathology in Parkinson's disease in all patients with Parkinson's disease, because all patients with Parkinson's disease have alpha-synuclein immunoreactive, alpha-synuclein containing Lewy bodies and Lewy neurites. So that was pretty much the beginning of a new era, and everybody started investigating in greater detail the alpha-synuclein, most of all from the neuropathological standpoint, following up of the work of Maria Grazia. And one of the neuropathologists who actually did it perhaps in the most systematic way was Heiko Brack. And what Heiko Brack did, he analyzed many brains, looking for alpha-synuclein pathology throughout the brain of these patients. And after all these analysis, he published another seminal paper in 2003, in which he described what is called now a Brack staging of development of Parkinson's disease. What he actually described in this paper is that alpha-synuclein pathology, so the Lewy bodies and Lewy neurites that contain alpha-synuclein, initially they start in the lower brainstem, and in particular in the medulla oblongata. Then they take a sort of an upward trend of diffusion, and you see the pathology moving rostrally, so from the medulla to the pons, and then to the midbrain, and ultimately the pathology affecting the entire brain of patients. So Brack basically described this very well. It described these stages very well, and you can see this is a table that is taken from this original paper that he published in 2003. And I have to say, you know, it's interesting for people, for all of you doing research, it's interesting to see how seminal paper do not necessarily have to be published or are published in super jumbo impact factor journals. Neurobiology of Aging is an excellent journal, but definitely doesn't have the impact factor of Science or Nature and so on. But the fact is that sometimes something that is really important and revolutionary is difficult for people to digest. I'm sure, and Heiko Brack sent his original paper to high impact journals, and there was a lot of resistance in accepting this concept of alpha-synuclein pathology evolving in stages and evolving from the lower brain to higher brain region. But this is, in fact, this finding has been confirmed and followed up by many other neuropathologists. And nowadays, it is believed that indeed this staging and this upward progression of pathology in the brain of patients with Parkinson's disease occurs in a good majority of patients, with, of course, some important exceptions. One thing that I would like to emphasize, again, despite the fact that this was incredibly novel and important, is that to be noticed the fact that one of the areas brain region that seem to be affected first by alpha-synuclein pathology is in the medulla oblongata, and in particular neurons in the dorsal motor nucleus of the vagus nerve. The other thing that also, and this we will come back to this important concept. And the other thing is that, as you might notice, the disease progresses over probably many years from the lower brainstem to the pons, and then finally reaches the midbrain. This is where the substantia nigra is, which means that by the time that neurons in the substantia nigra are affected by alpha-synuclein pathology, by the time these neurons become the major problem for patients, because it's the death of these neurons that ultimately bring patients to the physicians because of their inability to control movement anymore. But the disease has already developed for many years. So by the time we see the phenotype in these patients, which is the symptoms, the motoric symptoms that we see, tremor, bradykinesia, rigidity, that are due to the involvement of the substantia nigra and the nigrostriatal system in this pathology. By that time, the disease has already evolved rather significantly for a significant number of years. So this was already pretty exciting and interesting and new. So alpha-synuclein spreads throughout the brain and affect ultimately the entire brain, including cortical regions. But Brack went also one step forward, and he actually proposed and show evidence suggesting that not only, again, there is this ascending trend of alpha-synuclein pathology from the dorsal motor nucleus of the vagus nerve to nuclei in the pons, and then in the midbrain, and then in other regions of the brain. But he also actually proposed that perhaps the pathology may actually start in the peripheral nervous system, and in particular in the enteric nervous system, in the gut. And of course, this was an interesting hypothesis, and the hypothesis postulated that maybe the pathology starts in the gut and reaches the brain through the vagus nerve, and the vagus nerve, of course, ends up in the dorsal motor nucleus of the 10th nerve. But of course, this was not simply an hypothesis, it was also correlated by interesting observation. And of course, the main observation was the fact that indeed, Brack, as well as many other people, started looking for alpha-synuclein pathology in the gut. And indeed, you can find pathology in the gut, as it is shown in this image that I took from one of Brack's initial publications. Here you can see a nerve fiber of the Meissner's plexus in the stomach wall that is filled of alpha-synuclein, and it's clear pathologically affected by alpha-synuclein accumulation. So alpha-synuclein evolves in terms of pathology throughout the brain, but also it might evolve from the peripheral nervous system to the central nervous system. Once again, very new and very different from what we had thought at the time. So let me then summarize some of these concepts, and let me tell you why somehow you can say that there was in PD, in Parkinson's disease history, there is a pre-alpha-synuclein and a new PD, a Parkinson's disease that has been significantly affected in terms of its pathogenesis, in terms of its pathology, and so on, by the discovery of alpha-synuclein. So, and these are the differences from pre-alpha-synuclein PD and what I call the new PD, but the PD that is also informed by the discovery of alpha-synuclein. From the pathological standpoint, the pre-alpha-synuclein PD, as I've mentioned already at the beginning of the presentation today, was very much nigrostriatal-centric. We were really focusing on the nigrostriatal system, the dopaminergic system, and with the discovery of alpha-synuclein, these changed completely. We realized more than we had even imagined before that the pathology of Parkinson's disease affects the entire brain, and also, as I just mentioned, it may actually involve the peripheral nervous system. So a major change in our understanding of the disease. But these are other important changes that have been brought about by the discovery of alpha-synuclein. So, for example, from the clinical standpoint, it's not that we for clinicians, for neurologists, for us seeing patients, it's not that we didn't know that there were symptoms beyond the motoric dysfunction that characterizes patients with Parkinson's disease. But now with the discovery of alpha-synuclein, these non-motor manifestations of Parkinson's disease made more sense, because now we knew that the pathology underlying the disease invest and involve the entire brain. So now, generally speaking, we can make a distinction in saying that yes, it is true that because of the death of neurons in the substantia nigra, the dopamine depletion, we have the manifestation of motor symptoms such as tremor, rigidity, bradykinesia, loss of postural instability, and so on. But there are a constellation of other symptoms that affect patients with Parkinson's disease that involve psychological symptoms like depression, hallucination, psychosis, sleep disorders, sensory problems, autonomic dysfunction, and also cognitive impairment, including dementia. So all this constellation of symptoms that we see in patients with Parkinson's disease, with the discovery of alpha-synuclein, make more sense, because we can correlate to a disease that is actually a whole brain, whole body disease. Then from the etiological standpoint is also very important, because I mentioned at the beginning, most of all after the discovery of MPTP, everybody was talking about the environmental exposure, toxicant exposure, and so on. Now, with the discovery of alpha-synuclein, we realized that there is obviously a genetic component, since not only a single mutation in a specific gene, the one of alpha-synuclein, can cause the disease in families with this mutation. But then more and more studies have identified many genetic risk factors that increase the risk for the disease, for developing the disease. And these are other important changes that have been brought about by the discovery of alpha-synuclein. Some of these will be discussed later on. For example, we will focus on models that have been developed as a consequence of alpha-synuclein pathology. The biomarkers, which is also an important issue, the search for biological markers of the disease. Now with the discovery of alpha-synuclein, we are now looking at different markers than the previous one that were mostly focused again on the loss of dopaminergic neurons in the nigrostriatal pathway. So once again, the discovery of alpha-synuclein has been, I would say, revolutionary from the point of view of the definition of the disease and the understanding of the disease. And of course, we all hope that this will ultimately translate into something that we can do therapeutically also to improve the disease in patients with Parkinson's disease. But then, of course, some critical question arises when you have new discoveries, such as the discovery of alpha-synuclein. And one of the key questions is what makes alpha-synuclein toxic? Why alpha-synuclein, which, by the way, it's a protein that is normally expressed in our brain. So we all have alpha-synuclein in our brain. However, under certain circumstances that we don't necessarily understand yet completely, alpha-synuclein becomes the culprit of pathology. So what can make alpha-synuclein toxic? Once again, we don't know a lot of we don't have a lot of knowledge to address this question, but we have some interesting clues. One clue came also from genetic studies. I've already mentioned that single point mutations that change the protein in terms of a different protein, so qualitative changes of the protein, they can cause Parkinson, a form of familial Parkinsonism. But interestingly enough, a few years after the discovery of alpha-synuclein, of this single point mutation, geneticists also identify multiplication mutations that are also associated with the development of familial Parkinsonism. What does it mean? It means that in this family, there is no qualitative change in the protein, is that this individual, they do have more alpha-synuclein. And in particular, for patients or family who have duplication of the alpha-synuclein gene, they would have about 50% more protein or RNA, whereas patients with triplication of alpha-synuclein, as I think is shown on this um, graph on the left, they would have twice as much alpha-synuclein in the neurons than in a normal control. So, and interestingly enough, I'm showing this because families with duplication and triplication, and I believe these are pathological observations in patients with duplication of the alpha-synuclein gene. They do have a pathology that very much resembles what we see with idiopathic Parkinson's disease. Here you can see the loss of pigmented neurons in the substantia nigra. These are again classical Lewy bodies that you can see within neurons in the substantia nigra, but also Lewy bodies in neurons like in other areas of the brain, such as the locus coeruleus. And these areas of the brain are also very much affected by Lewy bodies in idiopathic Parkinson's disease. So bottom line is that patients with multiplication mutation have pathologically all the features that we see in Parkinson's disease. And therefore, we can conclude as a clue to the question, what makes alpha-synuclein toxic? Well, having too much alpha-synuclein makes alpha-synuclein toxic. And it's almost like having, unfortunately, human transgenics, i.e. individuals that have duplication or triplication of the gene, they have a lot of protein expression, and they do develop Parkinsonism. So this is one clue that we have in terms of what makes alpha-synuclein toxic. Another clue to this question came also a few years later. And now, once again, let's go back to what we briefly also discussed and saw a couple of days ago. You remember we talked about embryonic transplant, fetal cell transplantation. There was developed also in the MPTP model that Aisha described a couple of days ago. And I also told you that some of the MPTP patients also, they underwent embryonic transplant, fetal transplantation. So again, 10 years later, more or less, after the discovery of alpha-synuclein, these important two papers came out back to back in Nature Medicine. And one of these papers described this case. This was a 61-year-old woman who had an history, of course, of Parkinson's disease. And she, in 1993, she was transplanted bilaterally with fetal mesencephalic tissue. And as a result of that, as we discussed this already a couple of days ago, she had tremendous improvements in her symptoms. Before the transplantation, not only she was very much severely affected by Parkinson's disease, but also she was not very well responsive to the medications that are given to people in patients with Parkinson's disease, levodopa. And in fact, she had dyskinesia and off symptoms, and that's why her only chance in a way was to try this transplantation, this fetal transplantation, and she did extremely well after that, to the point that in 1997, for example, when she was of course monitored throughout the years, but she was doing very well. She had no off time. In other words, she was always responsive to treatment. She only had mild dyskinesias. And all these lasted about 10 years after the transplantation. And then in around 2004, she started experiencing worsening of her Parkinson's disease. And she continued to deteriorate during the next few years, and she died of cardiac arrest in 2007. What I would like to notice is that, again, 2007 is 14 years after the operation for the fetal transplant. So the brain of this patient was analyzed very carefully, and the first thing that was noticed, this is the striatum, and you can see that tyrosine hydroxylase staining in the striatum. It really shows that the grafted tissue was very much viable. You can see these tyrosine hydroxylase immunoreactive neurons. So there were still neurons that had been grafted in 1993 that were still surviving and innervating the striatum of this woman, which was interesting. But the thing that was perhaps interesting and also very surprising was this. When they looked and they stained the brain of this woman for alpha-synuclein, they did see alpha-synuclein inclusions in the substantia nigra. This is the substantia nigra tissue stained for alpha-synuclein, and this is the same tissue stained for ubiquitin, which is another marker of Lewy bodies and Lewy neurites that is often used to stain the tissue of patients. So in the substantia nigra of this patient, as you might have expected, there were alpha-synuclein inclusion. The thing that was not expected is that they also found inclusions in neurons in the grafted tissue. So this is, again, fetal tissue that was grafted into the striatum of this patient, and 14 years later, in these neurons, you see Lewy bodies and Lewy neurites. So how do you interpret all these? One possibility is, of course, that these neurons that were grafted into the brain of this patient, even though, again, they were healthy, young neurons because they were coming from fetal tissue, they were somehow affected by a neurodegenerative process around them that ultimately made them develop Lewy bodies. But the other possibility that was postulated right after that in this paper was that perhaps alpha-synuclein from tissue around the grafted tissue invaded and jumped from neuron to neuron, and ultimately this misfolded alpha-synuclein entered the neurons in the graft and they made them develop the Lewy bodies. So as a result of this interesting observation, people started looking at the ability and the property of alpha-synuclein to propagate from neuron to neuron. And in fact, now it is thought that one of the mechanisms or one of the properties that make alpha-synuclein toxic is this ability to jump from one neuron to another. And perhaps because of that, alpha-synuclein can also jump from one region to another region in the brain and spread, as Brack has described, from one region to another in the brain of patients with Parkinson's disease. So these are two important concepts: enhanced alpha-synuclein expression and neuron-to-neuron propagation. So let's go back to this idea of developing models. So when you develop a model, you want to start from some basis and say, well, if we think that increased expression of alpha-synuclein is important, and if we think that neuron-to-neuron propagation of alpha-synuclein is important as a pathogenetic process in Parkinson's disease, we would like to create a model in which to study these events that, once again, in which we have enhanced increased alpha-synuclein expression and neuron-to-neuron propagation. So this is one of the things that we started thinking a few years ago when we thought of developing a valuable new model for investigating alpha-synuclein pathology. But we also thought of something that it would have been even nice to complement to this concept. So enhance alpha-synuclein expression, neuron-to-neuron propagation, but also Brack told us that the propagation of alpha-synuclein starts in patients in the dorsal motor nucleus of the vagus nerve. So could we develop a model that somehow capture these three clues that we have about alpha-synuclein and the development of alpha-synuclein pathology? Enhanced expression, propagation from neuron to neuron, and involvement of the dorsal motor nucleus of the vagus nerve. So these were the questions. Could we overexpress alpha-synuclein in the dorsal motor nucleus of the vagus nerve? And if we do that, does this alpha-synuclein propagate? Does it spread as it does in patients with Parkinson's disease toward more rostral brain regions? So how did we do that? Well, we thought, and in a way, when you do these experiments, you really don't know if they're going to work or not. But we thought that one way to reach neurons in the dorsal motor nucleus of the vagus nerve would be through the vagus nerve itself. You can see here that the vagus nerve is a nerve that connects to the dorsal motor nucleus of the vagus nerve. So what we thought was maybe if we inject viral vectors into the vagus nerve, these viral vectors will travel through the nerve, reach neurons in the dorsal motor nucleus of the vagus nerve, transduce these neurons, and make these neurons synthesize a lot of alpha-synuclein. So we tried to do the experiment, and the first question was, can we obtain transduction and targeted transduction of the dorsal motor nucleus of the vagus nerve or the lower medulla oblongata by injecting into the vagus nerve? And the answer was, fortunately for us, was yes. This shows, for example, a sagittal image of a rodent brain in which you see that after injection of adeno-associated viral vectors into the vagus nerve, you see that specifically alpha-synuclein, and in this case it's human alpha-synuclein that is expressed in the rodent brain, is expressed in the lower brainstem, in the dorsal medulla oblongata. And in fact, if you do kind of three-dimensional reconstruction after doing clearing of the tissue, you can see these are neurons in the dorsal motor nucleus of the vagus nerve. And these neurons, they overexpress a significant amount of alpha-synuclein as a result of transduction through AAV injected into the vagus nerve. So the first answer to the question was yes, we can indeed induce overexpression of alpha-synuclein, increased expression of alpha-synuclein, specifically in the dorsal motor nucleus of the vagus nerve. Then, of course, the following question was, once we have this increased expression, is alpha-synuclein going to propagate, to diffuse, to spread toward more rostral regions of the brain? And the answer actually is yes. So when we inject into the vagus nerve, we overexpress alpha-synuclein in the dorsal motor nucleus of the vagus nerve and in the dorsal medulla oblongata. If we wait a few weeks, you start seeing that alpha-synuclein within axons in region rostral to the lower medulla oblongata, in this case in the pons. And then if you wait longer and longer, you see these spreading of alpha-synuclein, caudorostral spreading of alpha-synuclein. And this is schematized in this slide. At 4 weeks, these experiments were originally done in the rat brain. After 4 weeks, you see there isn't so much of alpha-synuclein outside the medulla oblongata. But if you wait a few more weeks, you start seeing alpha-synuclein in the pons, in the midbrain, and ultimately after several weeks, you start seeing alpha-synuclein in 4 brain regions. So finally, I think we addressed our 2 fundamental questions and say, do we have a model in which we can have overexpression, increased expression of alpha-synuclein, specifically in the dorsal motor nucleus of the vagus nerve, and can this lead to spreading of the protein toward more rostral brain region? And the answer was yes. So as much as you can be happy with developing a new interesting model, the importance of the model is not the model itself, but is what you do with it. So, and of course, in our case, as it is in most of cases, what we want to do with our models is to investigate mechanisms, to understand how these, in this specific case, how this propagation, how this spreading of alpha-synuclein occurs, and what affects the spreading of alpha-synuclein in this model. So we have been developing hypotheses and experiments to evaluate different mechanisms. And in the next 2-3 slides, I will show you some of the results that we have obtained that I think may be interesting in terms of providing, again, clue that could be relevant to understanding pathogenetic processes in Parkinson's disease. One of the first mechanisms that we thought would be worth investigating was oxidative stress. And for many reasons. One of the main reasons is that oxidative stress has long been hypothesized to underlie pathogenetic process in Parkinson's disease. So the question that we decided to address or to investigate was what does oxidative stress do in our model of alpha-synuclein spreading? So to address this question, and this is why, as we were discussing a couple of days ago, it's always interesting to keep in mind previous models and maybe to adapt the previous model to the new models. And so what we did, we thought we can induce oxidative stress in the brain of these animals by injecting paraquat. You remember we talked about paraquat 2 days ago as one of the toxic agent that is able to induce nigrostriatal degeneration. But the question here is what's different. Does injection of paraquat by inducing oxidative stress, does it change the spreading of alpha-synuclein? So we did the experiment, and this is just to show you what we do when we do these experiments. You can actually evaluate quantitatively the spreading by counting the number of fibrils that you see that contain human alpha-synuclein, pathological fibers containing human alpha-synuclein in the brain of animals, of rodents. And each of these dots here, this is a section, a schematic section of the pons. Each of these dots here represent a single neuron, a single axon. So you can count these, in several animals and get an average. So this is what we see, what we saw in this experiment when we injected the animals with saline, control animals. But then, of course, the key experiment was to see what happened when we injected animals with paraquat, and these are the results. As you can see that when the animals were exposed to paraquat, there were much more, the number of axons containing alpha-synuclein in the pons, as well as in the caudal midbrain and the rostral midbrain, was significantly enhanced. So indicating clearly that an oxidative stress promotes the spreading of alpha-synuclein, promotes the passage of alpha-synuclein from neuron to neuron. And this is, we can see this schematically here. You can see the neuron-to-neuron transfer is a property of alpha-synuclein of likely pathophysiological relevance. And when we induce an oxidative stress, we do see that much more the protein is exchanged and between neurons, and much of this protein goes from neurons, for example, in the dorsal motor nucleus of the vagus nerve to neurons in more rostral brain regions. And what I have indicated in red, which is something that I will not describe in detail because but if you want, we can discuss later on. We also think that the oxidative stress modifies alpha-synuclein and generates species of alpha-synuclein that have a higher propensity to travel from neuron to neuron. So this was the results of this initial set of experiments. Now I'll show you another interesting set of experiments in which we follow up this concept. If oxidative stress is capable of promoting alpha-synuclein spreading, where this oxidative stress may be coming from. And generally speaking, and this is also in particularly true for the paraquat model, a major source of oxidative stress can be microglia. And microglia, and in particular, this enzyme called NADPH oxidase in microglial cells. This is a multimeric enzyme with several subunits, and you can see some of these subunits normally are present, they are in the membranes, such as the GP91 and the P22 subunit. These are the three subunits, they're usually in the cytosol under normal conditions. When this subunit, when the microglia are activated, this subunit reach the other subunits at the level of the membranes, and as a result of that, the entire machinery comes in place, and basically this enzyme reduces oxygen and starts generating superoxide as a result of microglial activation. So the question that we wanted to address was, is it possible that microglia play an important role in the effect that we see, in other words, in the fact that we see increased spreading of alpha-synuclein as a result of oxidative stress. So how did we plan, or how did we design this experiment? What we thought of doing was to do the experiment in wild-type animals, in normal animals, so saline and paraquat in normal animals, but also to do the same thing, saline and paraquat injection, in animals, in knockout animal for this subunit of the NADPH oxidase enzyme. In the absence of this subunit, NADPH oxidase cannot function properly. It will not generate superoxide. It will not contribute to oxidative stress. So we decided to make this comparison. We did the experiment. So we injected saline and paraquat into wild-type animals, controls, and then we injected saline and paraquat to animals that didn't have a functional NADPH oxidase. And these are the experiments, the results of this experiment. This is what I have already shown. This is what happens in the wild-type animals. You have, you can count the fibrils in the ponds as well as other regions, but and then when you inject the animals with paraquat, you see that there is a significant increase in the number of these fibrils, indicating that paraquat, as I've shown before in the previous experiment, that oxidative stress induced by paraquat increases the spreading of alpha-synuclein. But then we repeated the same experiments in knockout animals, and the results are dramatically different. In these knockout animals, as you can see, there is no effect of paraquat. There is no increase of spreading as a result of paraquat administration, indicating that we were able to prevent oxidative stress and the effect of oxidative stress by blocking the production of superoxide and other reactive oxygen species in microglia, indicating once again the relevance and the importance of oxidative stress in these processes, and indicating the important role that microglia may have in promoting this oxidative stress and ultimately facilitating the spreading of alpha-synuclein from the dorsal motor nucleus toward more rostral brain regions. So I want to conclude this and then leave to Aisha to continue the presentation and the talk on models, and also showing some data that we have obtained in some of these models. So this is our working hypothesis based on some of these results, as well as results that I didn't have time to show you. So first of all, as I mentioned before, when we have oxidative stress, alpha-synuclein is transferred more actively, more efficiently from one neuron to another, and potentially species of alpha-synuclein that have a greater propensity to go from one neuron to another are generated and maybe underlying this effect. But one of the interesting things to us is that in order for alpha-synuclein to be transferred from one neuron to another, it needs to reach the extracellular space. So there is alpha-synuclein that is released from neurons and is in the extracellular space. And we believe that this extracellular alpha-synuclein may have in itself deleterious consequences. And one of these consequences could be that alpha-synuclein could bind to specific receptors on microglia and induce microglial activation. Once microglia are activated, then they will start producing more and more reactive oxygen species, kind of generating this vicious cycle in which you have oxidative stress. This starts this neuron to neuron passage of alpha-synuclein, but then ultimately feeding itself through a mechanism that generates even more oxidative stress through microglia. Once again, this is still a working hypothesis. And by the way, I would like to mention that we're also working on other mechanisms that could be relevant to the spreading of alpha-synuclein in the model that I've described to you, with the idea ultimately perhaps to be able to see how can we stop these mechanisms for doing what they do, and ultimately how can we reverse these, and hence to see if this can ultimately translate into developing therapeutic agents or therapeutic strategies that could be applicable for patients with Parkinson's disease. And I will stop here, and I will stop sharing my screen and leave the screen to Aisha. And maybe the best thing to do would be to for you to wait until the end of Aisha's presentation, unless you have really urgent questions, and then at that point we can further discuss some of this data. Thank you very much.
All right, so if there are no urgent questions, I'll continue, but I can also ask once again, does anyone have any questions to Dino? Then I will share my screen. Okay. It's visible, right?
Yes.
Yes.
Mm-hmm.
Yes.
Okay, so I will continue to my talk following Dino's presentation, starting from the discovery of alpha-synuclein and following the presentation that I've done 2 days ago on the models that reproduce nigrostriatal pathology. And in this case, last time I talked about the toxin models. Today I will continue and talk about other models of alpha-synuclein pathology that leads to alpha-synuclein expression. And I will repeat some of these models that I have discussed 2 days ago. That kind of induces not only nigrostriatal pathology, but also alpha-synuclein upregulation and aggregation, and talk about a bit about transgenic approaches, the knock-in models and overexpression models of alpha-synuclein, and how well they recapitulate alpha-synuclein pathology. Apart from transgenic, although gene delivery is also some sort of a transgenic approach, but I will talk about viral vector mediated delivery of alpha-synuclein as a model of alpha-synuclein overexpression, and also talk about the seeding model, where Dino briefly described about this the spreading of alpha-synuclein that perhaps also a little bit of what Brock described, that there might be a pathogenic species or alpha-synuclein itself that induces seeding of alpha-synuclein pathology that might also cause the spreading of the disease. And based on this hypothesis, there are models that reproduces alpha-synuclein pathology. So just to follow up on the model that I've described before, the MPTP model that I told you, that's a systemic injection of MPTP in non-human primates and mice leads to nigrostriatal neurodegeneration. But that is not the only pathology that is seen in this model. Although MPTP model was described and developed before the discovery of alpha-synuclein, when researchers later on went back and looked at alpha-synuclein in this model, they already saw that after MPTP injection, so in this model, this is a single acute injection in squirrel monkeys, that already 1 week later there is increased endogenous alpha-synuclein mRNA in the brain. And after 1 month, there is still alpha-synuclein upregulation in the brain, and one can see the accumulation of alpha-synuclein in the substantia nigra of these animals 1 week and 1 month later. Later on, some other researchers also followed up on this topic and found that MPTP indeed induces alpha-synuclein aggregation in other species of non-human primates, and also this leads to so upregulation of alpha-synuclein leads to also pathology in the brain of non-human primates. So MPTP is not only the only, it's not the only toxin model that leads to upregulation of alpha-synuclein. In fact, the herbicide paraquat, the one that Dino talked about, and I also introduced its effects on the nigrostriatal system, that it caused dopaminergic neurodegeneration. But that is not the only thing that paraquat does. Increased oxidative stress in a chronic fashion, or a double dose injection of paraquat, also leads to increased alpha-synuclein in the substantia nigra. Also, the rotenol model, this is, as you remember, is a mitochondrial complex inhibitor, OXPHOS regulator. It also leads to alpha-synuclein accumulation in the substantia nigra. So it is clear that these toxins that are inducing nigrostriatal pathology does not only causes dopaminergic cell loss, but these mechanisms of increased oxidative stress or mitochondrial dysfunction also leads to upregulation of alpha-synuclein. That what Dino showed, that in patients with higher alpha-synuclein, increased alpha-synuclein in the brain is also a pathologic mechanisms that may cause the loss of dopaminergic neurons. So we did mention bits that alpha-synuclein is not specific to the nigral system, and it does not cause accumulation. So Parkinson's disease is not limited to the substantia nigra. So when these toxins are injected systemically, it's at that time when the disease was more nigrostriatal centric, researchers focused on the substantia nigra. But actually, what happens is that when these toxins are injected, it does not only cause alpha-synuclein pathology in substantia nigra, but in fact, it's when, for example, this is an example of rotenone. oral treatment of rotenone in wild-type mice, it also causes alpha-synuclein upregulation and accumulation of alpha-synuclein in the gut. And this accumulation does not remain only in the gut, but eventually it leads to alpha-synuclein accumulation with time in a progressive manner in the brain. So these images are taken from the dorsal motor nucleus of the vagus nerve in the brainstem, where Braque describes that is the initiation site of Parkinson's disease in the brain. So you can see that accumulation of of alpha-synuclein, and this is as compared to the control animals over time, also leads to accumulation of alpha-synuclein in the brain. And what I'm not showing here is that these animals also show some motor dysfunction. So clearly, when the pathology is initiated in the body with systemic administration of toxins that causes mitochondrial dysfunction, this pathology might eventually lead to Parkinson's disease. This is another example. Just to note that when this time paraquat is injected in animals that overexpress alpha-synuclein. So here in this experiment, the animals already had underlying alpha-synuclein pathology. But here what this experiment shows that when the animals are treated with paraquat orally, systemically, the alpha-synuclein pathology is initiated and even progress further in the gut. So here, this is phosphorylated alpha-synuclein staining in the gut of animals so C is here control and E is exposed. So you can clearly see that, yes, the control animals, because of their alpha-synuclein overexpression, already have some alpha-synuclein pathology, but treatment of paraquat, in other words, increasing oxidative stress systemically in the whole body, leads to even more increased pathological alpha-synuclein accumulation. These are the photos from the gut, and this pathology also eventually increases the pathology of alpha-synuclein in the brain as well. So let's move to more genetic models of alpha-synuclein. So one can immediately think that, and this is also what researchers have thought about after the discovery of the mutation of alpha-synuclein and the discovery of alpha-synuclein, that is the main the protein that accumulates in Lewy bodies. Researchers thought that, okay, so if we overexpress alpha-synuclein, we will develop Parkinson's disease models, because at that time, researchers immediately thought that this might be the cause of Parkinson's disease. So beginning of 2000, several groups developed alpha-synuclein overexpressing animals, knock-in models, either using wild-type alpha-synuclein, or at that time the mutations of alpha-synuclein were already discovered, or mutated forms of alpha-synuclein. So what typically these models reproduce is a very strong alpha-synuclein pathology that even in the toxin models that I showed you, that there is expression of alpha-synuclein, accumulation of alpha-synuclein, the pathology with these models is not that strong in terms of alpha-synuclein accumulation. But these animals typically features very strong alpha-synuclein aggregation with post-translational modifications that are also observed in Lewy bodies, which I will come to this, but one of the most typical post-translational modification that is seen in Lewy bodies is the phosphorylation of alpha-synuclein. So these animals typically show alpha-synuclein aggregation in the brain, and they are post-translationally modified, meaning that they're phosphorylated, they're truncated forms of alpha-synuclein is also there. Ubiquitination is also typical in these animals within alpha-synuclein expressing neurons. And some of them also show neuronal loss. But the problem is that because most researchers have used ubiquitous promoters, the alpha-synuclein expression in the nigral neurons is not that strong, and cell loss is not always seen in dopaminergic neurons. What's also typical, that is also kind of new to alpha-synuclein models that was brought with these models, is that there is a progressive pathology that mostly we don't see in toxin models. In toxin models, for example, MPTP would be one of the most progressive one, that one can see changes already in 1 month. But these animals show alpha-synuclein aggregation and accumulation when they start aging. So these animals actually reproduce quite nicely the aging process of Parkinson's disease, the aging aspect of Parkinson's disease. Some of them, at least, they show motor symptoms, not always dopaminergic, dopamine neuron related, but there is some outcome measure that one can measure the functional dysfunction in the functional outcome in these animals. And some of them also show non-motor symptoms that in Parkinson's disease, that is of course typical, that not all symptoms are motor. Patients suffer gastrointestinal dysfunctions or sleep disorders or olfactory deficits, that some of these animals with alpha-synuclein pathology in the brain can nicely show some of these non-motor symptoms. Here I have shown, I try to summarize the extensive number of mouse models, transgenic mouse models of alpha-synuclein overexpression, because there are many of them available, and they have researchers have used different promoters. Ty1 promoter, prion protein promoter are the very typical ones that are commonly used, that show relatively strong alpha-synuclein pathology. So here you can see that the brain aggregation in this wild-type human alpha-synuclein expressing Ty1 mouse model starts already at 4.5 months. And depending on the mutation, there are different progressions. What you can here see is that these animals do not show any nigral cell loss. There is also the prion promoter models that also do not reproduce cell loss, but they have progressive alpha-synuclein pathology in the brain. The only models that reproduce nigrostriatal neurodegeneration are the ones that express mutated alpha-synuclein with the synuclein promoter, or with the TH promoter, the ones that express truncated forms of alpha-synuclein that show after 2 months loss in substantia nigra. So this is actually very strong model of transgenic animals. So these models, as I said, they are very useful in reproducing alpha-synuclein pathology in the brain, but in most cases, they perhaps, rather than Parkinson's disease, they could be referred to as synucleinopathy models because they're not necessarily nigrostriatal specific. So from this, I will also introduce you to the gene delivery of alpha-synuclein that are most often used with adeno-associated viral vectors. There are also models that were mostly used in the past using lentivirus or adenovirus, but AAV vectors became very popular in neuroscience field because they are relatively clean models. They are safe because they're not pathogenic to humans, and they do not cause a lot of inflammation by themselves. They do not integrate to the gene, so in terms of experimental safety, but also other safety reasons, they are considered safe. They do not cause mutations in the gene of the animals that you inject. And yeah, they are very widely used for in vivo gene transfer. So what we typically use in our lab is adeno-associated viral vectors that overexpresses human alpha-synuclein or mutations of alpha-synuclein. This is an old study showing some of the features of this model. So here the virus is stereotactically injected directly into the substantia nigra. So this is again quite nigrostriatal specific, but it's other as an advantage, they also reproduce the alpha-synuclein aspect of the disease. So what you can see here is in A, it's a tyrosine hydroxylase staining from an animal that was injected on only one side, that you can see that the striatum already has lost tyrosine hydroxylase staining. The terminals are degenerated, as well as the cells in the substantia nigra on the injected side. This is alpha-synuclein staining, human alpha-synuclein staining, that you can see that once injected into the substantia nigra, it affects the nigrostriatal pathway and leads to here pathological forms and pathological axons in the striatum, as well as neuronal loss in the substantia nigra, which is summarized here. So here, this is a count of neurons in the substantia nigra from animals that express either human alpha-synuclein or GFP as control. And this is an experiment that we have done in our lab that you can see compared to the GFP, there is a strong cell loss in the substantia nigra after 8 weeks of injection. Okay, so I will come back to the viral vector models to show you some examples of what we do in the lab and also some other features of the model. But what also became very commonly used the last 5 years is the preformed alpha-synuclein injection model, a preformed fibril model of alpha-synuclein. So this work was pioneered by Virginia Lee's group and Calvin Look as the first author, where they injected fibrils of alpha-synuclein. So this work kind of followed up the findings of the cell transplantations, where researchers have seen alpha-synuclein accumulation in the transplanted cells, and the hypothesis at that time is that alpha-synuclein spreads from one cell to another. And another thing that they also hypothesized, that this is some sort of a prion-like spreading, and it requires seeding of the alpha-synuclein that is expressed in the cell. So based on that hypothesis, Calvin Look and Virginia Lee prepared fibrillar form of alpha-synuclein and injected them into the striatum. Once these animals were, so these are mice, once these animals are injected into the brain, what happened some time later, 30 days or 90 days is the examples that were given here, is that these animals start forming endogenous phosphorylated alpha-synuclein accumulation in the brain. And interestingly enough, it's not only in the injection area, so these animals are injected into the striatum, but the accumulation was in the striatum as well as in the cortex, also in the substantia nigra, in the thalamus, in the cortex. So the areas that project to the striatum also has taken up alpha-synuclein. This is another study. I put this, there are many studies showing the reproducibility of this model. I chose this because this was done in rats, just to explain that the model is not species specific. One can reproduce this in rats as well as in non-human primates, and it does not necessarily need to be with preformed fibrils, but Lewy body extracts also give similar results. So if one extracts Lewy bodies from patients and injects them into the brain, they also lead to pathology and prion-like, or let's call it seeding in this case. They also seed endogenous alpha-synuclein pathology. Here you can see that upon injection, so this is a bilateral injection, with time, at 6 months, these animals show severe cell loss in the substantia nigra, and also this model is also featured with increased inflammation with time. So Brock hypothesized that the disease is not only seen in the brain, and it might start from the gut. And based on this hypothesis, researchers, Brock themselves as well, start looking into the peripheral nervous system and start reporting quite some pathology also in the periphery, but mainly around the vagus nerve and vagus nerve connected regions. So based on these observations, researchers have also thought that, okay, so if we inject these fibrils, these preformed fibrils in the gut, do we see or induce pathology in the brain? And the answer is, interestingly, yes. When these animals, so these are mice, wild-type mice receive mouse preformed preformed fibrils into the gut, to the stomach wall and in the duodenum. With time, researchers start observing phosphorylated alpha-synuclein accumulation, first in the dorsal motor nucleus of the vagus nerve and in the brainstem at 1 month time point. But with time, at 3 months, 7 months, and 10 months, one can appreciate the increase. So these red dots represent alpha-synuclein accumulation, phosphorylated alpha-synuclein accumulation. And one can see that there's a progressive pathology in the brain. And this pathology also specifically leads to nigral cell loss, again, in parallel to the pathology, the increased pathology progression. So I told that I will come back to the viral vector model. So since some of these models can reproduce the spread from the gut to the brain, and when I say some, I showed you the preformed fibril model, but also at the beginning of the talk, I did mention that, for example, rotenone treatment, oral rotenone treatment also leads to a pathology that starts in the gut and eventually goes to the dorsal motor nucleus of the vagus nerve and leads to accumulation of alpha-synuclein in the brain. So there is clearly evidence showing that an initiation of pathology in the gut leads to pathology in the brain. But is it only one way? And would one also see spreading of pathology from the brain to the gut? So this is an experiment that we have done some years ago in our lab that, as Dino described, we inject human alpha-synuclein expressing AAVs into the vagus nerve, and we see expression of human alpha-synuclein in the dorsal motor nucleus of the vagus nerve, so this is the brainstem, but also in the nodose ganglion on the vagus nerve, which, as you know, vagus nerve projects to the guts, to the stomach. And of course, we see human alpha-synuclein expression once we inject AAVs into the vagus nerve in the vagal terminals in the stomach wall. So there is nothing very surprising here. But what we wanted to know is that what happens if we inject AAV vectors expressing human alpha-synuclein into the brain? So not in the brainstem, but more frontal to the midbrain, and would one see alpha-synuclein in the gut? So this is the experimental design that we have done. we injected human alpha-synuclein AAVs into the ventral midbrain containing the substantia nigra. And the question is whether alpha-synuclein here would travel to the dorsal motor nucleus of the vagus nerve, which I also need to stress the fact that dorsal motor nucleus of the vagus nerve does not project to the frontal brain. So it projects only to the vagus nerve. So the only way that alpha-synuclein will be taken up by these neurons is that if alpha-synuclein passes from one neuron to another. And what we ask is, do we see alpha-synuclein in the terminals of the vagus nerve? So this is a general description of how alpha-synuclein expression looks like when we inject alpha-synuclein into the brain. And as you can see that injection is done here to the ventral midbrain, covering substantia nigra. And of course, this expression leads to very nice specific expression of alpha-synuclein in the terminals of dopaminergic neurons in the striatum, as well as in other dopaminergic neuron projection areas. And also some projections that also is taken up that goes to the caudal midbrain. But what's, I don't know if you can see from here that this is the brainstem, and this white area in the middle is the dorsal motor nucleus of the vagus nerve. It's white because there is no, there is no well-known described connections between the dorsal motor nucleus of the vagus nerve and to the midbrain neurons. And so these neurons only project outside of the brain. And indeed, when we inject alpha-synuclein expressing viruses at 2 months time points, the dorsal motor nucleus of the vagus nerve is relatively empty. There is no human alpha-synuclein or very little fibers passing from here and there. But with time, we start seeing accumulation of alpha-synuclein expressing projections to that region. So, of course, seeing the projections does not mean that the neurons of the dorsal motor nucleus of the vagus nerve picked up this alpha-synuclein. And indeed, when we do a double staining of choline acetyltransferase, which marks the cholinergic neurons of the dorsal motor nucleus of the vagus nerve, and alpha-synuclein, we see alpha-synuclein accumulation in these neurons and in their projections at 6 and 12 months time point. And indeed, alpha-synuclein, the vagus nerve enters the brain from the side and connects to the dorsal motor nucleus of the vagus nerve. And one can also stain this entering nerve bundle at the level of the brain. And here you can see that at 6 months and 12 months time point, we start seeing human alpha-synuclein along the vagus nerve in these axons. And the next question would be, how about in the periphery? Indeed, at 6 months and 12 months time point, but not before, we see human alpha-synuclein staining in the vagus nerve. And in these are the blue color are the enteric neurons in the gut. And the brown staining here is human alpha-synuclein. So these antibodies do not detect endogenous alpha-synuclein, but only human alpha-synuclein. So if one sees a specific staining like this staining can only come from the viral expressing exogenous form of alpha-synuclein. So here, I think it is quite clear that alpha-synuclein is a protein that pass from one neuron to another. And in fact, this passage of alpha-synuclein does not have to be from the gut to the brain, as other experiments have shown, but this traveling of alpha-synuclein can also be from the brain to the gut. So it's one can say that it travels long distance to both directions. And here, what's quite important is that there are many nerves in the brain, many projections, and alpha-synuclein specifically, when injected into the brain, is taken up somehow by the vagus nerve. So when alpha-synuclein is injected into the to the periphery, it is carried by the vagus nerve. And when we inject alpha-synuclein into the or express alpha-synuclein in the brain, it is taken from the brain to the guts. So there is an important role that vagus nerve plays in Parkinson's disease progression. So I think this is one of the important things that our work shown, that there's a propensity of the vagus nerve and the dorsal motor nucleus of the vagus nerve to take up alpha-synuclein for some reason. Yeah, so here we detect alpha-synuclein in the stomach, which after injecting or after overexpressing alpha-synuclein in the brain, which also suggests that perhaps although most of the studies show that alpha-synuclein may travel from the guts to the brain, I think one should not exclude the fact that perhaps there might be a possibility that in certain subtypes of Parkinson's disease, maybe the genetic forms of Parkinson's disease, or these days Thomas Beach suggests that there is also a limbic predominant and a brainstem predominant Parkinson's disease, that perhaps Parkinson's disease does not necessarily start always in the gut, and one should keep an open eye of where the initiation sites could be. So this was the work that we have done in our lab. But to conclude this session is that so here we describe models that reproduce alpha-synuclein pathology, and they are not only useful to study nigrostriatal aspect of the disease, but with our new knowledge on Parkinson's disease, they also have the capability to reproduce the pathology that is seen in Parkinson's disease as a whole body disease. Although, for example, the alpha-synuclein overexpressing model that I have shown you looks like it's nigrostriatal specific, but one can, of course, also study the pathology of alpha-synuclein as if in means of injecting it to other brain regions, like the one that Dino described, that we injected into the vagus nerve to reproduce different aspects of the disease. What these models are quite useful is that they help us elucidate the pathogenic mechanisms, which could be used together with the toxin models. But here, what they very nicely reproduce is, other than neurodegeneration, is the fact that they show alpha-synuclein aggregation and Lewy body-like formations in the brain. And of course, spreading of alpha-synuclein with these models could be also studied quite nice, and the mechanisms could be studied with using these models. Apart from all these, I think in terms of translation of findings to clinics, they also reproduce a relatively slower progression compared to most of the toxin models, for example, compared to 6-hydroxydopamine model, where the neurodegeneration is achieved in 1 week. Here, depending on what model one chooses, there is a longer time for interventions that allows perhaps better time window, therapeutic window for therapeutic interventions. So I think this is it from my side. Thank you very much. We can now perhaps take some questions to this talk or for Dino's.
Thank you very much to both of you. I think it was a very clear and exhaustive presentation of both the toxin model and the first meeting, and now on the alpha-synucleinopathy models, especially with a focus on the spreading of the pathology, which is crucial. So I think that maybe we can also collect questions which may relate to the first meeting, if there are, or to today's presentation. Are there any questions? While we wait, I have a question on whether you detect some kind of peripheral phenotype in the mice which have been injected in the Niagara and which developed the gut pathology, the gut alpha-synuclein accumulation. I don't know whether you checked whether they may display some, I don't know, gastrointestinal-like symptoms.
Unfortunately, we did not check that at that time. So we have only pathology outcome from that work. But yeah, it's something that we are also considering, perhaps, to also get into that. It's quite interesting.
And then another question I have is about the kind of alpha-synuclein species. I don't know whether you characterize also a little bit the species which are in which you develop in these models.
Yeah, I think I actually we for obvious reasons when we became interested in investigating the role of oxidative stress, the obvious post-translational modifications of alpha-synuclein that we started focus on were nitration of alpha-synuclein, as well as formation of oxidatively modified alpha-synuclein. What I can tell you is that we, in vivo, we did consistently see a relationship between accumulation of nitrated alpha-synuclein and propensity of increased spreading, suggesting that the nitrated form of alpha-synuclein may be somehow acquiring a higher propensity to spreading. But the thing that was perhaps most interesting was that at that point, we did also an experiment in vitro, because as you can imagine, this is something that maybe we do have to throw on the table discussing about the PD model. There is only so much you can do sometimes with the in vivo models that we have been focusing on, but sometimes you also have to parallel these with in vitro models where some of these mechanisms can be evaluated with greater detail. So when we started focusing on a nitrated alpha-synuclein, we did also an experiment in vivo, which we did in collaboration with Sanjay Lee. They have an interesting model. It's a Venus system by which they express two parts of the alpha-synuclein molecule that once they get together in different cells, and only when the two parts of alpha-synuclein they get together, they get fluorescent. So now you have part of the cells that express part of alpha-synuclein, other cells that express the other alpha-synuclein. You co-incubate these cells and you start seeing how much of alpha-synuclein fluorescent is formed, indicating passage of alpha-synuclein from one neuron, from one cell to another. When we did this experiment, we saw the same thing that we saw in vivo, which means basically if you induce oxidative stress in this cellular model, you do see more fluorescence, you do see more synuclein getting from one cell to another. But the thing that was very important, I think, and intriguing to us was that if you do these experiments in vitro and in the incubation medium, you add antibodies against nitrated alpha-synuclein or against nitrotyrosine, which is basically the part of the protein that is nitrated, all these effect is completely abolished. In other words, these antibodies of course, the antibodies do not enter the neurons. They mostly work outside the neurons. So they will block the protein in the passage from one neuron to another. So when you do these experiments in vitro, you block completely these fluorescence formation and indicate this cell-to-cell passage of alpha-synuclein by adding antibodies specific for nitrated alpha-synuclein in the medium, suggesting or actually providing, that was, I think, the first clue that we had, that nitrated alpha-synuclein could indeed be a species of alpha-synuclein that acquires a higher propensity to go from one neuron to another. And of course, there are interesting implications about that, because as you can imagine, everybody's now trying to develop a way to stop alpha-synuclein pathology using, for example, antibodies against alpha-synuclein, or using silencing alpha-synuclein expression and so on. I think it will be very important, in my view, to identify specific species of alpha-synuclein that might be more easy to target without targeting alpha-synuclein itself, and might be more effective to target for therapeutic intervention. Perhaps one of these species is nitrated alpha-synuclein.
Yes, thank you for your response. I fully agree with this, and it's also in line with what we are seeing in our models. And I would also add that you know, now there is the novelty of the polymorphs. Who knows whether the nitration impacts diversely impacts on the conformation? We don't know. So it also needs to be explored, I think.
Absolutely.
Deeper details.
Yeah, and the other species could be oligomeric alpha-synuclein, because we do also have evidence that the oligomeric species might also be more easily transferable from one neuron to another.
Yeah, absolutely.
Yep.
Thank you.
Thank you.
Are there more questions from the young? There is two questions. Do the knockout that Prof. Di Monte described specific for the different NOx subtypes, nitric oxide, and is the knockout global or cell specific? And so this is the first one.
I think it's a very important question, because there is a form of NADPH oxidase that is not specific for microglia. For those experiments that we did not look to distinguish between the microglia and but we believe it's mostly the microglia form, because that's the one that in the case of paraquat seems to be particularly active in promoting the formation of superoxide. But that's something that can be done using more tissue-specific knockdown of the NADPH oxidase. Yep, thanks.
So the second was on can you also please comment on the role of other brain-resident macrophages in driving ROS production in PD, since in AD, perivascular macrophages are shown to be major sources for ROS production that drives tau hyperphosphorylation. This is
yeah, I can start, and maybe Aisha, if you want to add. I mean, you might recall on Thursday, a couple of days ago, I showed you some data. The interest in microglia and neuroinflammation, it's something that it goes back decades. As you might recall, I showed you data in MPTP patients suggesting that in this patient, after the initial insult with MPTP, the reaction of tissue mediated by the immune reaction, the microglia, seemed to be also perhaps continuing over time and becoming problematic. So there is no doubt that also in Parkinson's disease, microglia and neuroinflammation play an important role. And then, of course, there is this link between microglia and the generation of reactive oxygen species, which I think it was, once again, something that was heavily investigated a few decades ago with the MPTP model, and now is becoming, in my view, also based on the some of the experiments that I've shown you and some of the experiments we're doing in the lab, continues to be a more and more intriguing link between microglia, oxidative stress, reactive oxygen species, and alpha-synuclein pathology. So that's it's clearly something that we should pay attention more and more. The other issue, however, and if you don't mind, I'll take a few more. The issue with this, the neuroinflammation and this process is that how do you convert this into therapeutics? Because the big promise about neuroinflammatory drug is that, as you know, if you go to any kind of pharmaceutical companies, they have on their shelves tens of potential anti-inflammatory drugs. So why we have not been able to convert this knowledge into actual therapeutics? That's a fundamental, difficult question. Also because aside, there is evidence also from epidemiological studies that individuals who, for whatever reasons, take anti-inflammatory drugs throughout their life, they might have a reduced risk for Parkinson's disease. So there is evidence from the epidemiological side, there is evidence from the basic research. How is it that we are not developing? I think part of the problem, and again, this could be a completely different lecture, but part of the problem is that unfortunately, we might intervene too late when we might hypothesize giving this drug to patients too late in the disease process to have an impact by acting on microglia, by acting on some of these targets, including ROS formation. So the big goal over the next years will be also to be able to link this study on pathogenetic mechanisms to study on biomarker, identify early on biomarkers of the disease, and maybe intervening as early as possible might help also to see how much this agent can be helpful in preventing the progression of the disease. That's but again, there might be also other important factors involved in this interesting relationship between microglia, neuroinflammation, and PD development.
Yeah. Other questions? Maybe one of you can comment on the strategies which are under development, which have been introduced by Dino, to clear alpha-synuclein from the brain.
I can give you some, once again, Aisha, you can chip in after, but I think this is an interesting approach, which is also something similar has been done in Alzheimer's disease with amyloid, tau, and so on. So I think it seems like a logical thing to think of. Let's get rid of the protein that is bad. Now, I still think it's not a bad strategy necessarily, but there are several important considerations. One of the first consideration is let's be careful, because to get rid of alpha-synuclein might not be on the long run a very, very good strategy, because this protein is in the brain, is actually, I think it's been calculated that is almost less than 1%. It is a significant protein expressed in the brain. And that means that even though we still don't understand exactly all the functions of alpha-synuclein, it might not still be a good idea to get rid completely of alpha-synuclein. About 15 years ago, we began doing experiments using siRNA to block and to the alpha-synuclein, to get rid of alpha-synuclein expression. And we did some experiments in non-human primates. And I remember that at the time we were working with a pharmaceutical company, and my and the position of some other people in research working on this was, let's use an siRNA that does not completely deplete alpha-synuclein. You can choose something that depletes alpha-synuclein by 50% or so. So we did some initial work doing that, and we were successful in showing that, at least in non-human primates, we were able to decrease of 50% alpha-synuclein in the substantia nigra, and this didn't seem to have any negative side effects. Unfortunately, then I moved here to Germany, and I wasn't involved anymore in the subsequent part of the study. But I know that the big temptation has been to go back to, we always like to say, the more the better. So, and I think, unfortunately, sometimes that shouldn't be done. So my point first in general is not to eliminate alpha-synuclein for the brain. The second thing is perhaps to figure it out what is the component of alpha-synuclein, specific species of alpha-synuclein, specific aspects of alpha-synuclein pathology that could be more precise target than in terms of preventing the elimination of alpha-synuclein while blocking the bad alpha-synuclein. I keep saying sometimes to people that alpha-synuclein is kind of a Giano Bifronte, it's a schizophrenic personality. It's a protein that has important function, but then for reasons that we still don't understand exactly why it becomes pathological under certain conditions. We can guess some of these situations could be, you know, for example, alpha-synuclein is heavily expressed during the development. But interestingly enough, during development, alpha-synuclein is also expressed under conditions in which in the neurons, there is plenty of chaperone molecules. So perhaps under those conditions, there's a lot of alpha-synuclein, but there are also chaperones, and then might counterbalance the expression of alpha-synuclein. It's very different when you start overexpressing alpha-synuclein in an adult brain, aging brain, when maybe these counter mechanisms are not in place anymore. So once again, I think these are good strategies. The antibody strategy is something that has been started a few years ago. Several companies have been generating antibodies against alpha-synuclein. I haven't seen, even though theoretically some of these initial studies should be finished, I haven't seen any results supporting. There is experimental research supporting that these the antibodies might be able to block alpha-synuclein pathology. I haven't seen anything in humans yet.
I think the Spark, the one from Biogen, was stopped.
Yep.
Has been stopped. While there is another one, the Prothena Pasadena
Yep.
study, which is still going on, but just because they found a device to monitor the patients during the daily living, so they could see some kind of improvement.
Yep.
But you know, then we miss I mean, a biomarker or a tracer to see whether there is an actual reduction. So
you're absolutely right. But you know, the thing that again, a few years ago, when I was just a bit perhaps more naive about alpha-synuclein, I thought that the strategy with alpha-synuclein would be interesting because let's face it, the problem that we have with patients is not so much, or not only the fact that patients come to the doctors at the beginning and they have motor dysfunction and so on. The problem with these patients is the progression of the disease. So naively, I've always thought maybe if we want to stop nigrostriatal degeneration, yes, we need biomarkers and intervene as quickly as possible and so on. But maybe if we get something that stop or slows down this progression, even at the time when the patient shows up to the doctors and has a few, although not nice, but a few motor dysfunction symptoms, if we could stop the disease there. I mean, if you go to any patient and you say, if you go about 5 years in terms of your symptom, would you, I mean, they will be jumping up and down. And that's why I thought initially maybe this gives us an opportunity to intervene even during the disease process, as long as we manage to stop the progression of the disease. Easy to think, very maybe optimistic view, more difficult to realize. But, you know, I was even thinking sometimes and fantasizing about the possibility of targeting alpha-synuclein siRNA or antibodies or whatever to key regions that, according to the Braak staging, seems to be at the in the intercept of all these different pathways. I don't know, targeting the amygdala, targeting some of these areas of the brain that's theoretically, if you could stop it there, the progression and the propagation of the pathology might stop. Again, easy to say, much more difficult to do, but perhaps this might still be the case. I mean, we all hope that, again, this alpha-synuclein has given us this opportunity. And if Braak is right, if all this work with progression and with the spreading is correct, we might be able to develop something that may not necessarily reverse the disease, may not, but it might halt the disease and the progression of the disease. And that would be a tremendous achievement for patients, of course, in itself.
Yeah. I think one of what's missing with the clinical part of the research is also perhaps that the patient's progression is mostly assessed by the UPDRS scoring. So their response to dopamine, their motor symptoms, and there are very little tools to understand whether any of these antibody treatments, chaperones, or whatever that we think of targeting alpha-synuclein has indeed worked. So perhaps it starts working and maybe will slow down the disease, but it's very hard to tell within the clinical trials time. So they're mostly done much shorter, because the shorter, the cheaper the clinical trial is. And there are, for example, very little that we can do in terms of imaging alpha-synuclein in the brain, for example. There are, yeah, so I think one of the major issues is that perhaps it will, in 5 years or 10 years, the treatment might even help the patient not to develop, I don't know, perhaps on-off fluctuations or other dopaminergic related side effects. And but I think the outcome measures are very much focused on the nigrostriatal integrity of the disease. I have seen a couple of studies that are developed that are designed to also detect perhaps some outcomes in terms of non-motor symptoms. But we have very little idea about how much the progression of the non-motor symptoms correlate with the motor symptoms. I think there are some gaps between connecting the preclinical study, the animal models, and the outcome of the translational of the clinical studies in terms of what to expect in terms of recovery, slowing down the disease, and the time point of how this information is going to be translated and predicted by the clinical research.
Yeah, absolutely, it's true, and I think that there is also a gap with the animal studies, because not always people look at other symptoms rather than the nigrostriatal associated symptoms, I mean the motor symptoms. So we don't have yet a clear idea of how we have to evaluate the novel treatment also, I think.
Yeah, it's absolutely true. But having said that, I would also say that this is one of the also very important things that discovery that we have seen with alpha-synuclein. To give you an example, more and more, and this somehow follows up on what Aisha is saying, but also somehow say we are moving in a direction of going away from simply evaluating Parkinson as a motoric dysfunction. One of the most promising way of designing clinical trial now is, for example, to identify a population of very high risk of converting to Parkinson's disease. For example, REM sleep disorder. People who have REM sleep disorder, a significant proportion of these individuals will ultimately develop a Parkinsonian syndrome within a relatively short period of time. So now more and more clinical trials are being developed looking at these non-motoric symptoms to evaluate whether or not we can have an impact on the conversion of these individuals from just simply having no REM sleep disorders to also having Parkinsonism. So slowly but surely we are moving in that direction, and but there are obvious problems in general that we cannot overcome very easily. The progression of the disease is not something that you can often, even with the UPDRS or whatever measure you have, even if we had necessarily a good biomarkers. It's not something that you can change or reverse in a matter of 6 months, 1 year, 2 years. So, and as you all know, organizing a clinical trial with any kind of agent for 5-year period is pretty much would bring most of the pharmaceutical company or biotech company bankruptcy. So somehow we need to use our judgment and use some of these new criteria that we have developed. Again, REM sleep disorders, maybe certain population. For example, now we are working with clinicians to see whether or not individuals who have GBA mutations, they do convert some of them to the disease. Can we identify this subpopulation of individuals that have a high predisposition to develop a Parkinsonian syndrome and use these for a more effective and rapid evaluation of therapeutics? And so we are moving in that direction. Hopefully we will have the problem with these studies is that the high probability of failing, and every time you fail there is a rebounding effect in driving people more and more away from becoming more pessimistic, becoming less willing to give it a try. But that has to change, because sometimes, again, you need to do what happened in the 60s with levodopa. You need to have the gut to do something different, new. And sometimes, even knowing exactly what you're doing, you might have the right answer. And that's what perhaps is going to change significantly everything from the therapeutic standpoint.
Yeah, that's true. Also, there is the complication of the levodopa therapy as well. Yeah, and it's not really
absolutely
a small problem.
Not at all. And as I said a couple of days ago,
it's great to have levodopa, but then.
Yep. And of course, there is the pharmaceutical company. They also have interest in pushing drugs. So there is a lot of dopamine agonists and so on. But the reality is that the best drug continues to be levodopa. And the dopamine agonist, when sometimes people say the dopamine agonist, they don't give you as much side effect as levodopa. Yes, they don't, because they are not as good as levodopa. So when you start developing some of these agents, you only have to say, well, you give a dopamine agonist, you see less dyskinesia. Yes, you do, but you also see less of effectiveness. So you also see less dyskinesia if you give less levodopa. So I think this has been somehow a misleading message that has been somehow given also from the research standpoint. Levodopa indeed is the best drug that we have, has these side effects. We can, I think the best thing from the therapeutic standpoint is on a patient basis, very individualized patient basis, is to titrate the levodopa treatment or treatment with dopamine agonist to the point of preventing as much as possible the development of these dyskinesias, and minimize the effect of that. But frankly, I started doing research on Parkinson's disease a couple of years ago, and we were talking about dyskinesia as one of the best things we could have solved. And a few years later, here I am talking about dyskinesia as one of the most difficult mystery to be solved from the pharmacologic standpoint. And less people, and in this, perhaps alpha-synuclein has not helped, because it has brought us away from the nigrostriatal system and from some of the two things that are exciting, new, spreading, prion, whatever. But at the end of the day, yes, I think if I had to say what could really help on immediately patients, I would say dyskinesia treatment. But that's not so much in fashion anymore, unfortunately.
We haven't mentioned that much, but some of these or most of the graft transplanted patients also develop some sort of a dyskinesia that is graft-induced dyskinesias, which without a proper dyskinesia treatment, you cannot reverse the dyskinesia. In L-DOPA treated patients, you can decrease the dose, you can stop L-DOPA treatments if the dyskinesia is severe. But you can't do anything. You cannot take out the graft anymore, and these patients will have dyskinesias. And I think it is very, very important if for example, if the iPS cell transplantation needs to go forward or if it becomes successful, one needs to really make sure that the dyskinesias are not severe, the graft-induced dyskinesias, but also perhaps in parallel start developing, if one cannot avoid dyskinesia development with graft transplantations, one needs to really focus on developing better treatments for dyskinesia treatments.
Yeah, that's true. That's really true. And implement also the dyskinesia model, because they are not really, yes, we have the good model of the 6-hydroxydopamine, but then we don't know whether we can find better ones yet.
Yeah, the problem with dyskinesia models is that in patients, dyskinesias develop in 5 years, 7 years, or 10 years, whereas our animals don't live that much. I think in MPTP, dyskinesia models are, I mean, I don't want to sound pessimistic, but they are probably the best with 6-hydroxydopamine model after 3-4, if it is a full lesion, after 3-4 L-DOPA injections, the animals become dyskinetic. How well does that reproduce? Yes, of course, there is some sort of model that one can work with, but I think it is a very, very severe dyskinesia that whatever you try to treat, it does not work. Perhaps it's not because it won't work in patients, but perhaps the dyskinesia model with 6-hydroxydopamine is very severe.
Okay, are there any other questions? No. If not, I think I have to thank you again very much for these wonderful presentations and for the very insightful discussion. And I hope we will have the occasion to repeat something like this in the future, because I think it was really good, especially for the students, to have this kind of event. We have been stopped for a long time in doing seminars and all of these activities, so I'm really happy that in the end we could finalize this organization.
Yeah. And hope that next time it could be face-to-face.
Yeah, absolutely.
But this is also fine. I enjoyed our discussion very much, and thank you very much.
Yes, we need to adapt to all the situation. Okay, thank you very much.
Thank you very much.
Bye-bye.
Bye-bye. Keep in touch. Bye-bye.
Bye-bye.